My name is Carmen Galan. I am the chair of Cost Action GlycoNanoProbes. The aim of this action is to bring together experts in the area of glycosciences from all around Europe to develop the next generation of functional glyconanomaterials for the development of diagnostic tools and targeted therapeutics. Carbohydrates, proteins, lipids, and nucleic acids are the biomicromolecules that constitute the fundamental building blocks of life. Among those, carbohydrates are key players involved in a wide range of molecular recognition events, from protein folding, cell communication, bacterial and viral infections, to fertilization. Cell surface carbohydrates can differ considerably between cell lines and also between healthy and diseased states. These differences can be exploited for the development of early diagnostic tools, prevention and/or the treatment of diseases via, for example, molecules and probes that can target and interfere with those interactions between carbohydrates and its receptors. However, despite their biological significance and therapeutic potential, these important biomolecules have been investigated a lot less compared to nucleic acids and proteins. The vast complexity of carbohydrate systems, combined with the scarcity of glycan-based tools for study, have been a major challenge in glycobiology. Therefore, the production of tailored and structurally defined glycan-based probes for biomedical applications has represented a significant advancement in the field. On the other hand, nanotechnology provides a new array of techniques and platforms to study glycosystems. Recent developments in the field have provided access to an advanced toolkit of synthetic nanomaterials and the technique to study such molecules at high resolution. In order to successfully develop new glycan conjugated or carbohydrate-derived materials, interdisciplinary collaborations between material science, chemists, biologists, immunologists, microbiologists, as well as medics, is important. And this is what this network has been trying to accomplish over the last 4 years. We have established a functional network of researchers from academia and industry, bringing together experts from specific centers of excellence from different subjects such as materials, synthesis, nanotechnology, biomedicine, microbiology, cancer research, from 31 different countries across Europe, including 16 countries that fall under the ITC umbrella. We have over 260 members, and throughout the action we have organized events and activities to facilitate the exchange of ideas, access to infrastructure, and the know-how. We have provided training and mentoring for graduate students and early career investigators by a training school, a special early career dedicated workshops, and our short-term scientific mission. During COVID, we faced many challenges. We couldn't meet face-to-face, and instead we organized virtual events such as workshops where we allow participation, particularly of early career researchers, so they could present their work, and also more established academics. And in this manner, we managed to stay connected. Where possible, we still organize small events where people could meet following the COVID guidelines of each individual country. And overall, we were able to maintain our connectivity and our relationships. We're very proud of what we have accomplished thus far, and we hope that the relationships and the networking opportunities that we have developed will continue beyond the lifetime of this network. We're very grateful to COST for funding us, and we look forward to the future. Thank you.
We have organized meetings, schools, and workshops where students, academics, and early career researchers have been able to present their work, network, and establish new collaborations. We have created leaflets and posters and dissemination video where we talk about how we work together in the glycoscience to develop new treatments, diagnostics, and glycomaterials. You can find more about our activities at our webpage GlycoNanoProbes.eu and social media.
My name is Cristina Nativi and I am co-chair of Cost Action 18132 and since the second run period, grant awarding coordinator. Short-term scientific missions are one of the pillars of a cost action, and GlycoNanoProbes has been committed in granting as many short missions as possible. In this plot, it is reported an overview of the short-term scientific missions accomplished per grant periods. Globally, GlycoNanoProbes has supported 42 young researchers to perform short-term scientific mission and 3 others to attend international conferences. It is immediately evident the reduction of these activities during the second grant period due to COVID restrictions. But COVID did not reduce the interest in scientific exchanges and did not stop short mission for too long. Indeed, grant requests started over in the third and fourth grant periods, and we have been able to grant 11 missions in the third period and 11 in the fourth grant period. 14 different countries have been involved in hosting young researchers granted by GlycoNanoProbes. Spain hosted the majority of them, 9 in total, followed by Ireland, which hosted 6 young fellows. Young researchers coming from 16 different countries were financially supported by our cost action, and as reported in this slide, 9 of them came from Portugal, 5 from Italy and 5 from Hungary, 4 from Czech Republic, 4 from Turkey and Ireland. Well, thanks to short-term scientific missions accomplished in the framework of GlycoNanoProbes, new collaborations have been established and many others reinforced. Collaborative papers have been published and others are under preparation. So we can conclude saying that, like for multivalency, together we are the strongest.
Hello, my name is Andrea. I'm 25 years old and I am a PhD student in organic chemistry at the University of Florence under the supervision of Professor Cristina Nativi, and I will soon start my second year of PhD. As a member of the Coast Action GlycoNanoProbes 18132, this year I applied for a short-term scientific mission and I was granted for a split mobility. So at the beginning I spent 3 weeks in Lisbon at the Universidade Nova de Lisboa under the supervision of Professor Filipa Marcel. In Lisbon I focused on MGL, which is a really important protein in our immune system, which is known to interact with cancer cells. Then I spent 1 month in Bilbao, where I enjoyed Professor Jesus Jimenez Barbero lab at the research center SIC Biogune, where I basically worked using different techniques to study the interaction between carbohydrates and protein. Thanks to Coast, I was allowed to meet new people and make new friendships. More importantly, I gained new multidisciplinary skills and I was able to extend my scientific network, which is a really useful outcome in a short-term scientific mission.
This short-term scientific mission looked at investigating the molecular recognition mechanism of glycan probes by the protein human macrophage galactose-type lectin through the use of NMR binding studies. The mission was a collaboration between the groups of Carmen Galan and Filipa Marcelo and took place in August 2023 at the Caparica campus of the Nova School of Science and Technology. This STSM set out to give me the opportunity to learn about and gain experience in different NMR techniques I had not used previously, as well as to understand how these experiments can be used to study the interactions between proteins and ligands. The protein studied was human macrophage galactose-type lectin, or NGL, and the glycan probes were synthesized in our lab in Bristol for testing of their binding to this protein. Human macrophage galactose-type lectin is a calcium-dependent carbohydrate-binding protein, or lectin, that has been widely studied in the Marcello research group. It is known to specifically bind to glycan residues that contain terminal N-acetylgalactosamine moieties. The protein is expressed on dendritic cells and activated macrophages, and plays a role in modulating the immune response to infections and cancer, making it an attractive therapeutic target. One NMR experiment used in this SDSM was saturation transfer difference NMR. This works by irradiating the spectrum at a region containing no ligand signals, which allows saturation to transfer through the protein via spin diffusion. When a ligand is bound, this saturation can be transferred to the protons that are in close contact with the protein. SDT-NMR uses the difference between a spectrum taken when the protein is selectively saturated, the on-resonance, and a spectrum where the protein is unsaturated, the off-resonance. The difference spectrum shows only the signals of the ligands that receive saturation transfer from the protein, as the intensity of the signals in close proximity to the protein will decrease in the on-resonance spectrum. These experiments showed us the important sites in the ligands we tested, and therefore new ligands can be designed based on this, with the aim of achieving high affinity binding. Another NMR experiment used was proton-nitrogen heteronuclear single quantum correlation NMR, or HSQC. These experiments are protein observed and show the backbone and side chain amine NH bonds from the protein. The chemical environment of each amino acid defines the position of the signals, and this environment changes with differing pH, temperature of the signals, and also upon binding of a ligand. Chemical shift perturbation is observed on the residues of the protein binding site upon ligand binding, and this was used to observe binding to glycan probes. Overall, binding studies were performed combining the protein NMR expertise present in the Marcello lab with the carbohydrate ligands synthesized in the Galan lab, and the binding of different ligands to the protein MGL was observed and compared. Succeeded in gaining insight into the binding of the protein MGL through studying the interactions with glycan ligands. The results of
Cancer is one of the main problems that humans faced. It's causing millions of people die each year, and it's decreasing the life quality, and the treatment of the cancer is quite expensive, causes billions of dollars each year to the governments. And the best way to deal with the cancer is early diagnosis, to diagnose the cancer when it starts. There are different techniques, even though some of them are very successful, but the selectivity and sensitivity still are not in the desired level. Let's assume this is a cell, and particularly a line cell. When it undergoes cancer, only certain things are changing on the cell. Its shape, size, appearance look quite similar with the normal cell. But when we look at the molecular level, certain things on the cell surface or even within the cell are changing. These are, let's say, lectins. Lectin composition on the cell surface are changing upon cancer development. Lectins are proteins specific to carbohydrates, and the specificity can be at antibody-antigen level. And this gives us an opportunity to target the cells to detect them nearby the regular healthy cells. To do that, in our study, we developed fluorescently conjugated carbohydrate gold nanoparticle conjugates. As you see here, carbohydrates can be ornated on the gold surface, which brings multivalency to target the lectins. And when we want to detect them, they will bind to the cancer cells. And these lectin compositions are not present, or maybe just minimum amount are present on the regular healthy cell. And when we try with the regular cell, like healthy cell lines, we see no binding. This means the carbohydrates, if they are designed for the cancer cells through the carbohydrate-lectin interactions, we can target them. This will not only bring us to detect, also this will help us to produce cargo units to carry the therapeutics to the cancer cells, selectively killing the cancer cells. And we believe that exploitation of carbohydrate-lectin interactions will make a big impact on cancer diagnosis and at the same time the treatment of the cancer.
Hello, my name is Francesca Cadamuro and I'm a postdoctoral researcher at the University of Milano Bicocca. Thanks to the cost-action GlycoNanoProbes, I have the opportunity to spend my short-term scientific mission at the E3S Institute in Portugal. In my research activities, I functionalize biomaterial to develop 3D SEM mimetics, functionalized with different glycans to study the glycosignature effect at the SEM level. In the glycobiology in cancer group of Professor Celso Reis in the E3S Institute, I have the great opportunity to test my matrix with patient-derived gastric cancer organoids. It was a very great experience where I learned a lot and share science with amazing people. Thank you to the Cost Action to give me this opportunity to improve my multidisciplinary skills and have the possibility to extend my network. And thanks to the Professor Celso Reis to host me in his lab, and to my supervisor, Professor Laura Russo and Francesco Di Cotra, to support my research every day. Thank you.
Hello everyone, my name is Carlos Lima. I'm a PhD student in Nova University in Lisbon, and I did a short-term scientific mission in the lab of Professor Carmen Galán in the University of Bristol. I think that this short-term scientific mission was very productive in many ways, because since I've done it, I realized how difficult my colleagues have it there. So doing the synthesis of the sugars for me to use. And also I think I could provide them with some more information about what do we do with them, and I think it was very nice for them as well, so they can understand what they are working for. Besides that, it was very nice to meet everyone, and it's better to know the people that we are working with, and it makes everything easier.